In this video, I'd like to describe to you two things. Firstly, there are some figures in Chapter 4 of Hancock describing generic signaling pathways. I'll go through each of those quickly just to explain my understanding and hopefully help you understand them. Secondly, I'll describe to you in a bit more detail a particular receptor and quickly show you how that receptor functions. And again, I've shown that in the lectures. So, quickly looking at Chapter 4. Chapter 4 presents a simplified pathway, and in this example we have a signaling molecule binding to a receptor. At the membrane, on the inside of the membrane, there are other factors which, when the receptor is activated, it interacts with these other proteins, and one result may be the activation of an enzyme activity which is embedded in the plasma membrane. Typically, this enzyme will instigate a second messenger which will propagate the signal into the cell, and this is a method of amplification. So this diagram shows an extracellular signal working with some components in the membrane, including an enzyme activity, to generate a second messenger. This second messenger is then able to interact with other proteins in the cytosol to activate those and then to affect cellular metabolism. In addition to activating a single pathway, often these enzymes can activate a complementary pathway so that the end result is a strong phenotype or a strong response to the initial signal. Now, in this example, it's a fairly linear, straightforward pathway with a branch point, but two complementary pathways are being activated. Now, signaling pathways are not always that simple, and some examples of divergence are also given in the textbook. So, for instance, this is a fairly similar kind of pathway, if you like. We have an extracellular signal binding to a receptor, activating an intermediary protein, which activates an enzyme, which activates some kind of second messenger. But also shown in the textbook is a divergent pathway which is also activated. So in this instance, we're activating another component of a signaling pathway to produce another signaling scheme. So an easy way to think about this is some kind of membrane component here being activated as well as a cytosolic component, and then the cytosolic component can be generated from this membrane component to activate some pathway. So it's still very generic and hand-waving, but at some point we'll give some real names to these proteins and look at some specific pathways. But we'll look at some schemes where there's a degree of divergence within a pathway to produce a secondary effect, and often these are complementary as well. Now, here we have an elaboration on the previous scheme shown here, in which more points of divergence have been highlighted. So in this instance here, each time a big red cross is indicated is a point where one or more pathways can be activated depending on the cell type. And typically this is a way that a single signal can activate different components of the cell, whether it be gene transcription or protein change in protein structure through covalent modification or something like that. And the figure given in the textbook for that is this one here. I've just done a little highlight here making things red for you, but basically this is the idea of multiple divergence, where many things are being propagated from a single signal. And the last example given is quite a nice one, and it's one we'll look at in more detail later in the course as well. Again, we have in this signaling pathway, we have the extracellular signal, we have our receptor, our membrane components being activated, but in this case, the activation of a membrane component leads to an almost single chain of events. And we'll look at this kind of pathway in more detail, but in this instance here, there's not a lot of crosstalk because there are helper proteins such as these scaffold proteins, which are keeping these proteins away from other components in the cytosol. And also, the close proximity of these proteins causes a very directed response. And typically, this kind of pathway leads to the activation of a transcription factor, which then turns on a bunch of genes. And typically, that bunch of genes have a similar function, but also many other functions may be captured within the genes that are activated. So there's a series of 4 schematic diagrams showing some signaling schemes without getting too bogged down in the actual names of these proteins and complexes. But we'll add some names to these things over the next few weeks. The other thing I just wanted to mention briefly, or show to you briefly, is this idea of how a particular signal can activate a particular receptor type. So that there's that the book talks about platelet-derived growth factor and about how it can activate a receptor. So I've kind of grabbed a figure, and I'll just work through a sort of a hodgepodge animation of the activation. So here we have the platelet-derived growth factor. It's a dimer of 2 peptides. I think they're covalently bound to form this this dimer here. Now, there's a receptor for this signaling molecule, and the receptor is typically plasma membrane bound. So here's the plasma membrane, and here's the receptor. Now, the receptor in this case consists of 2 polypeptide chains, which may not be brought together. So they're not close in space in the membrane. We've got to think of membrane as a fluid structure where proteins can move about quite freely. Now, the key thing here is that when the receptor comes into contact with the signaling molecule, the receptor complex forms, such as is shown in this diagram here. So here we have our signaling molecule, and it's bound to 2 components that make up the receptor and brought that receptor close together. The upshot of this is that within the cell, on this side of the plasma membrane, these 2 polypeptide chains have now been brought together. And it's the bringing together of those 2 polypeptide chains which is the movement of the signal from outside of the cell into the cell. So this is the signal transduction into the cell. Now, what does it matter that these 2 polypeptide chains have been brought together? Well, in this particular kind of signaling, and we'll talk about this signaling later, which is the RTK signaling, which is receptor tyrosine kinase receptors, and there's a whole bunch of these things. Now, each of these polypeptide chains has an enzyme activity, which is the receptor tyrosine kinase. It has a kinase activity. And when these 2 chains are brought together, it effectively activates the kinase activity as such. So here I'm showing the intrinsic kinase activity that exists within these polypeptide chains. When they're brought together, there's a conformational change. The tyrosine kinase activity is activated, and that is propagating the signal now. Now, the substrate for this tyrosine kinase is this polypeptide chain, and the substrate for this tyrosine kinase is this polypeptide chain. So what we get now is phosphorylation of the receptor by the receptor to give a phosphorylated receptor. So here I'm showing now a whole bunch of tyrosine residues on the receptor chain that have been phosphorylated. So we've had cross phosphorylation here to change the effectively to covalently modify these polypeptide chains. Now we have a case where we have two polypeptide chains that have been brought together by the signaling molecule. When these two polypeptide chains are brought together, we get the activation of the intrinsic tyrosine kinase activity. The tyrosine kinase phosphorylate the adjacent polypeptide chain, and that is the key because it presents now these phosphate groups which are the target for the binding of other proteins as such. So now we have other proteins that can bind to a phosphate group on a tyrosine residue. So we have instances of proteins such as, don't get too bogged down in the names, but here we have GRB2, we have RAS, we have phospholipase C, we have PI3K, phosphoinositol 3 kinase, and we have SHP2 and SARC. It doesn't matter what these protein names are, we'll deal with them later, but it's the ability of these proteins to recognize phosphorylated tyrosine residues, which is the next stage in signaling. So I've kind of chucked you in the deep end then and just gone looking at a receptor type in a bit of detail, but the important things to take home are the fact that the signaling molecule brings together the components of the receptor. It's the close proximity of these chains of the receptor on the inside of the cell which allow the tyrosine kinase enzyme activities to become active, and then we get this kinase activity which is phosphorylation. So we get phosphorylation of the chains, and then it's these phosphorylated chains that are recognized by these additional proteins which then will keep the signal moving along inside the cell. So I hope that's of some help to you, and good luck. Okay, thanks.